Hello and welcome everyone to Oconomowoc, Wisconsin. If you decide to click on this video and comment on it, be sure to hit the like button, subscribe for more videos. Yeah, whatever. Okay, so today we're going to be looking at insulin kinetics and how it relates to type 1 diabetes. So we have, let's get in the present mode first. That'd work. Present mode. Present. Present. Okay. So, insulin kinetics. We're going to be looking at a study out of Austria that by Malgorzata Wilinska. I'm not sure if I pronounced that right, and a bunch of other people with European sounding names um, who studied type 1 diabetes kinetics. And at the end, I found a couple of cool things about type 2 diabetes too, as well. So we're going to be looking at that. So, what is diabetes mellitus? Well, in Egypt, physicians in the 1550 BC noticed that people urinated frequently um, and that they were malnourished or seemed to have less energy. In the 1600s, so about 400 years ago, people decided to taste the pee of people who were had these symptoms, and they noticed that the pee was very sweet, like honey, and so that's what mellitus means, is honey sweet. Um, and and so I'll get into why that tasted sweet in a second. And in the 19th century, people, some scientists in Russia took out the pancreas of some dogs, and these dogs developed symptoms of diabetes. And during the Prussian War, they had to ration some food, and people who were affected by the rationing, their symptoms of diabetes mellitus, went away. Not completely, but partially. So, what is wrong when someone has diabetes? So, well, when a normal person or someone who's healthy eats food, it's broken down into glucose and then it goes into our bloodstream. And then insulin allows the cells to take in this glucose where it's stored as glycogen. So, people with diabetes lack the ability to put the glucose into their muscle cells or their liver cells. And so the glucose is trapped inside of the blood. And so when the glucose is trapped inside the blood, the kidneys have to work overtime to get the glucose out of the blood. And so what do? They put it in the urine, and the urine goes out of the body. And so when this happens, we take fluid from other parts of our body, and people become fatigued or thirsty or hungry. So people with type 1 diabetes, they cannot produce insulin at all. And these are the people that take insulin regularly. People with type 2 diabetes do this too, but they have type 2 diabetes for a different reason. It's not that they can't produce insulin, it's that the insulin is not effective in allowing the glucose into the cell. And I'll get into why that is later on. So, insulin kinetics. In type 1 diabetes, continuous and bolus delivery of rapid acting insulin. The rapid acting insulin is just a type of insulin that they use by Malgorzata Wilinska and others. So, in this study, the point of the study was to assess the physiological feasibility of 11 proposed models for the insulin delivery kinetics. They had 7 people come in, and they were postprandially monitored and treated with CSII, which is just a subcutaneous insulin infusion. And the postprandially means after a meal. So they were fed a meal, and then they were monitored, and they had insulin connected to them at a continuous rate. So the models that they proposed all had they each had an equation along with them, and the equation had different variables and these variables were things such as the delivery mode, whether it was in a bolus, which is just a one-time sort of large, or not large, it's just a one-time thing, and then the continuous delivery method, which is just over time it's added at a more at a continuous rate, like I said. So and then there's association rate of insulin to the insulin receptor and the fast pathway, slow pathway. And the local degradation is if the insulin just doesn't even make it into the plasma. I'll get into that in a second. So here we have the structure, I guess, of the proposed models. And so we have in the first column, you have the structure, the equation that for the model, and then what the equation describes, and here in the comments, and then the variables in the parameters section over there. So I guess just for this first four, they use pretty simple parameters here. The Q stands for just an amount of insulin. The U is input, and the K is obviously the rate the rate transferring from each one to the next. And so we see, I guess, for example, the rate at which 3 changes over time is equal to the rate at which Q3 in the first model here changes from Q2 to Q3, just depends on the rate at which the rate times the amount. So which is a pretty simple one. But then they started adding variables in the next ones we can see here. And they got pretty complicated as we keep going through the models that they proposed. For example, like I said, they proposed in 8-9 a faster channel where it doesn't go from one amount to the next. It just goes straight from the injection to the plasma insulin at a certain rate, which is 1 minus the rate from the top one. Let me see if there's anything else to point out here. On the next one here, we have the same thing, but like I said, there's local degradation at the injection site. What that means is the insulin doesn't even make it to the plasma. It is subtracted out. So we can see that in the equation here on the top. The LD stands for local degradation, and so it's subtracted off the equation at the end there. We also see the Vmax and the Km involved. So what were the results of this study? So this graph here simply shows the data points that they got from the people, the actual insulin concentration in their bodies, what was measured, and then these are the data points that they got. This second graph here is a weighted residual, which pretty much is just the standard deviation of how far each one of these models was from the data points they collected. So the models gave a graph and they fitted it to the data points, and this is how far away the model was to each data point. As we can see here, those were models 1 through 6, and this one is models 8 through 11. And we can see that model 10, the dark square, was the closest fitting model for the equation that they came up with for the data. And so for this graph here, they used the equation that gave the 10th model and put it over the data points that they got from the subjects in the experiment. And this is how close, I guess, the graph is to the data points. And so this also just shows how the scoring of which model was the best. On the right-hand side here, we have the a score calculated by an equation that they came up with, and these people were um, mathematical engineers, so the equation is a little bit more complicated than I could understand, but ultimately it just gave a score on wanting to limiting the parameters and how closely it fit the data. And so they got model 10 to be the best score, the lowest score. And so on this page here, like I said, the models gave us K values for going from the injection site to the plasma, the insulin in the injection site to the plasma. And so we see here that we have different K values for different proposed models of how it went from each place to the next. And I have an example here that I want to look at more specifically on the next slide here. We have monomers versus the dimers here, because the plasma, the insulin, excuse me, will sometimes form a dimer because of its structure, hydrogen bonding. And so the hydrogen bonding of the dimers will actually cause it to, according to this model, the rate will be slower going from the injury site to the plasma than the monomer one. So in conclusion, for this paper, they said that Model 10 was the best fitting model for the data that was attained. As I said, they had the scoring there system that they came up with to figure this out, and the residual, the weighted residual. And so this was the model that suggested that there was a local degradation, meaning just it's lost at the insulin. It just doesn't become usable at the injection site. So it doesn't even make it to the plasma. So that was for that paper. Now I'm going to move on to just, I guess, to wrap up here at the end, just a little bit of how type 2 diabetes differs from type 1 diabetes. So this figure right here from a New England Journal of Medicine paper that came out in 2014 details how insulin is rejected by the cell. So people eating a lot of fatty acid in their diet will have an increase in fatty acid in the plasma, which is taken into the cell in the form of triglyceride. And so that causes right here lipid droplets in the cell. So that means then the protein kinase C causes phosphorylation of the insulin receptor substrate S, or substrate 1, excuse me. And then the insulin is not able to bind the insulin receptor. And like I said, the insulin works as a key to the cell. If the insulin binds to the cell or binds to the receptor, the glucose can then come into the cell. But since the insulin's not binding to the insulin receptor, this causes the plasma glucose not to be able to enter the cell, and this causes the glucose 6-phosphate levels to be lower in the cell, which ultimately affects glycolysis pathways, which in turn decreases glycogen synthesis. And so this is bad because the glycogen is used by the mitochondria as energy. So there's no glucose being allowed in, so then glucose levels obviously raise in the plasma. So how do we fix this problem? On the next slide here, I have a couple of things that can fix this problem. And this whole new, this is a newer way of thinking of how to fix this problem. In the past, people thought, oh, you have a lot of glucose in your blood. What is broken down into glucose? Carbohydrates. So we need to lower your carbohydrate intake. And now they're starting to rethink this idea. So right now they're thinking well, since you have a lot of fatty acid in your cell, that's not allowing, if I go back to this slide, you have a lot of fatty acid in your cell that's not allowing glucose in. It's telling the receptors don't allow it in. You need to first get rid of the fatty, you need to use, your cell needs to use up that fatty acid in the cell to be able to start bringing in glucose. So you need to lower your fatty acid intake. You need to lower the protein intake, which also, as I'll show in a little video on the next slide, which also causes it to be higher glucose levels. And you need to eat more whole foods in a plant-based diet to reduce this insulin and tell your body to use up the fatty acid that's in the cell. So here's the video.
Yeah. So now, it turns out that dietary protein also has a very similar effect, independent of fat. So in some studies that were performed to try and understand exactly how protein affects your blood glucose values, what some researchers found was very eye-opening. So you see this complicated graph here on the left. Let's walk through it really slowly. On the very bottom, we have a curve which shows what happens to your blood glucose after you eat a low-fat, low-protein meal. So you see how your blood glucose rises and then comes right back down, okay? That's what's called a normal glucose response, or a normal glycemic response to a meal. If you simply increase the amount of protein in that meal, okay, you go from 5 grams of protein to 40 grams of protein, look what happens. Your blood glucose response goes up. We didn't touch fat. We only added protein. If you add fat and you take away the protein, you get a similar response. What happens if you add fat and protein together? If you add fat and protein together, you get that response. So what this shows you here is that by simply adding either fat or protein, and I'm talking considerable quantities of either one, you end up elevating your blood glucose more and more and more. If you have a meal that contains both fatty acids and protein together, your glucose is highest. It's a problem. There's story after story.
Okay, so that was, to me, was just very telling of what we have back here in this graph, or this figure here from the New England Journal of Medicine study. That shows that you need to get rid of the fatty acids that are in your cell first, and it's just a different way of thinking about it that's come up in recent years. So I hope you can learn a little bit of something today about the kinetics of insulin resistance in type 1 diabetes and in type 2.